Let's keep it very short. All right, let's get going. Welcome to the Condensed Matter / Biological Physics seminar, and this semester is benefiting from my leisurely sabbatical. So this semester I'm hosting a few speakers in a row, and one last week, as I recall, studying sort of magnetotactic bacteria. So I have another sort of bacterial biophysicist, Jing. I met Jing a few years ago in one of the New England complex fluid conferences. He was then a newly minted young faculty member at Yale. And I discovered at that time his primary training is actually soft matter physics. So Jing did his PhD at UIUC, training with Stephen Granick, working on sort of colloids, Janet particles. And then upon his postdoc, he went to Princeton, but accidentally got into a microbiology lab in collaboration with, Howard Stone, who was well noted for his work in fluid mechanics. So that's how got him lured in, but then he ended up doing work on cholera, Vibrio cholera, which is a nasty sort of bacteria, right? Everybody heard about cholera outbreak and stuff. So through this work, he has sort of transformed himself to be one of the actually very productive young scientists working in that. And so a few years, he basically got hired in the cellular and developmental biology department at Yale, and also a member of the Quantitative Biology Institute. Over the past few years on tenure track, he has done really a lot of impressive projects, and he's recognized for his, by National Institute of Health as sort of one of these sort of innovators. Got his innovation award by NIH. He also is a fellow of Bruce Wellcome Foundation, and then numerous other awards. And as you can really look in detail in his CV. But basically, I want students to kind of just hear his talk and the science and try to imagine what kind of landscape he has traversed. Somebody with physical science training, but now on the faculty in molecular biology. And then, so I want you to get some tips of the way he approached science. He's really, I want to say, a ball of fire. So we almost like, I have a number of experienced PhD students meeting with him one-on-one today, try to tie him up. So we'll see how much energy he has left. Hopefully not too much. We can still follow your talk. All right, Jing, go ahead.
Okay. Well, thank you, Jay, for the really nice introduction. I learned I still remain active. It's a long day because I'm going to talk about the active matter. Okay. So this could be a little bit different from what you were thinking, because in soft matter we talk about all kinds of active matter, but today I will be focusing on a particular form called biofilms. And biofilms are essentially surface-attached bacterial communities surrounded by the extracellular matrix. How many of you been to Yellowstone?
Many times for me.
Okay, many times. You might recognize this beautiful color. These are hot springs in Yellowstone. The reason why we like to go there is because of the beautiful color, which is actually produced by the pigments from the bacterial biofilms. Okay, biofilm is the major form bacteria survive in nature, but in our daily life, it's actually harmful, right? They can clog filters and pipes, and they can cause chronic infections that are very difficult to treat. Now let me change to another question. How many of you washed your teeth this morning? Okay, I hope all of you. The reason why you want to do this is because you don't want a biofilm to form on your teeth. So it could be a really bad thing. So speaking of bad things, the thing that we study in the lab are really nasty, but Vibrio cholera, which are these cute bacteria under the electron microscope that can cause a pandemic cholera. Okay, so if you want to know a little bit about the history of what's going on in the pandemic, this is really a nice documentary. It's not a science book, but it's written about the outbreak in Haiti. Actually, before COVID, the growth rate of cholera is actually the fastest in the history, only surpassed by COVID 4 years ago. So, there are different reasons there are many different reasons we like to study cholera in the lab. First, it has been suggested that biofilm formation is important for cholera survival in nature, in host, and during outbreak. And also, there are a lot of genetics tools that are available for cholera, so we can make all kinds of manipulations. So my lab will use a series of different approaches, ranging from imaging to material science to evolution and chemistry. So we're using this a integrated approach to study biofilm formation. And in the interest of time, today I'm only going to highlight what we did from imaging perspective, and then later connected to gene expression. So the question we're asking here is sort of developmental question, right? If we think of these biofilm colonies or biofilms, it is really the most rudimentary form of multicellularity, right? Of course, we on this side is way more complex. On the other hand, there are things that we can learn from biofilm in this very primitive form of multicellularities that can help us understand the physics behind. And also, I will also argue that it's also more environmentally adaptive, right? Anything go wrong in this process will be fatal, whereas in bacteria, it can grow in all kinds of environments. So the question we're trying to answer in today's talk is, how do biofilms reproducibly pattern themselves? What are the biophysical forces, and what is the role of gene regulation, and how these two couple together to define biofilms? So I know there might be some people interested in tissue in the audience, and I hope you can see some parallels here. So one thing that I've been doing since my postdoc is to develop techniques to watch biofilms cell by cell. Oh, by the way, if you have any questions, just raise your hand. I want this to be more interactive, okay? I don't have to go through the entire talk. Okay. So what you're seeing here is the one individual cholera cells that grow and expand. So they're growing. Go ahead, yes.
So do you have this bacteria in your lab?
We grow, so we're cholera lab. We have all kinds of pathogens.
If a student accidentally got it into their hand, then what happens?
You report to EHS. You wash your hand. There is a procedure, emergency procedure if you have.
So they are harmful.
They are we're BSA 2 level. So you just have to be, you have to wash your hand every time, and you don't drink it. But you have to wash your hand very carefully. If you get in there, you have to go through a procedure of washing off the hand and treatment, so on and so forth. But nothing has happened so far yet. Good. And the people are very careful about it. There are other the even nastier bugs in the lab that you have to be more careful about. It's not that bad. Like the infectious dose in cholera is pretty high. You really have to drink it to get infected. I hope that doesn't happen in the lab case. So, so back to the movie. What you're seeing here is how this is really what the biofilm we're talking about. We develop tools not only to image them, but also to segment individual cells so we can follow the position and orientation of each this rod-shaped bacteria over time. And from this, one thing you can immediately see is that you see all these dots here, right? They're not spherical cells. They're essentially a cell that has stand up on the surface. So when you take a optical cross section, they look like sphere. So these are postdoc works. So from that, we can come up with a refined model for biofilm formation. Cells swim around, they come to the surface, and they start to secrete this extracellular matrix. I have a big line of my research trying to understand the chemistry of the matrix, and I'm not going to cover that today. But you can imagine that once you have a few cells, you run into a problem, right? These cells want to grow and divide, but you also have neighbors which are also sticky to the surface. That generates pressure, and we showed before that there's instability for individual cells to verticalize. And now the problem solved, because now they send their offsprings in the third dimension until they grow these three-dimensional biofilms, in which the cells in the middle are all standing up on the substrate. Clear? Okay. But if you look at the picture again, there's one thing. Sorry, I don't know why it's so sensitive. One thing, other than the cells in the middle standing up, you also see this radial organization. It's beautiful. It's almost like a flower pattern. So that got us. This is not just one biofilm. If you look at all of them are radial in some way, right? So now we'd like to understand what is going on there, and it took us into a surprising path. By we, I mean my first postdoc, JP, who is now making big money in AstraZeneca. And the way we image the biofilm is that we grow the biofilm in submerged in water, but underneath a agar pad, okay? There are many reasons why we do that, but in this case, we have a flatter biofilm, so we have a bigger basal layer where we can quantify the radial organization. We also have beads in the hydrogel, so we can measure the strain and stress in the gel.
I just have a quick question about the standing model.
Okay.
So is the idea that once the biofilm reaches sort of like a fixed perimeter, as they keep dividing, the instability causes them to stand?
So the cells are attached to the surface, and then with the other is there is some stable configuration. But as the cells is growing, the pressure starts to accumulate in the middle.
And the reason the pressure accumulates is that because the perimeter can't continue
We have
expanding.
We have equations afterwards.
Outwards.
But essentially each cell can move, but there is a friction, right?
Mm-hmm.
And when the cells grow and expand, you always experience resistance from the peripheral cells.
Okay.
That's why the pressure is always highest in the middle of the biofilm. That's where the verticalization happens.
But this thing can keep expanding outwards.
It can keep
doesn't
expanding.
need to be
Because
fixed.
but verticalization releases the stress, right? Because it's when you verticalize, it changes the footprint to get smaller. That to release some of the stress.
Okay, thank you.
Great. So good. So we also developed tools so we can track and segment these individual cells. And I'm going to show this kind of plot a lot, which is actually a fan plot, right? You can see in the radial organization, you can see that the angle continuously changes as you go through the circumferences. The interesting thing is that when we remove the proteins responsible for cell surface adhesion, now immediately you have a random biofilm. Okay, the organization is gone. And if you remove both the cell surface adhesion and also you remove the extracellular matrix that cells secrete, the cells may get a little bit denser. There's no polymer in between the cells, but they're still disorganized. Okay, maybe locally two cells will be parallel, but overall it's pretty disorganized. Okay, so and we can quantify this orientation, right? So if you can define the radial order parameter, this is 1, this is -1. We can genetically control the bacteria such that we can dye in gradually more and more adhesive, and we found that the radial order increases as you increase the cell surface adhesion.
What does it mean to have a biofilm with no cell surface adhesion?
Biofilm, the bacterial secretes the extracellular matrix, but the matrix does not allow the cell to stick to the surface.
So it's just an aggregate.
It's an aggregate.
Okay.
So we do a lot of bacterial genetics in the lab. We actually solve the crystal structure of that adhesion, which is a different track of my lab. Okay. So yeah, so if you do E. coli or other non-biofilm forming species, you will see essentially cells are disorganized. So there's somehow there's special here. The question is how. And then at that time we formed this key hypothesis. Now the growth will generate pressure. And the pressure leads to cell verticalizing the core. And that is obvious. And what is not obvious is that, let me just read this. We hypothesized that these verticalized cells, because they don't grow in the plane, so they change the flow pattern in the plane, and that changing 2D profile causes cell alignment. So what does that mean? Well, in the pictorial way, what we're saying is that now here the cells wants to expand, and these cells don't are sticky to the surface, so they're verticalized. They grow in the third dimension. They don't contribute to the growth in the plane anymore, and that modulates the flow in the plane and leads to cell alignment. Well, I know number 3 and number 4 is hard to understand, but let me just go through this slowly. Okay? So the first thing we want to do is we want to actually look at the flow in the material. By flow, there's no actually cell motion. Okay, everything is due to growth and division. But we need to track the flow. We can't really put a tracer particle, right? So what we do is that we put the tracer particle in the cell. So what we do is we engineer a string in which each cell is carrying a bright puncta. Okay? Sorry. Let me just use I'll put this movie. You can see how this works. We have each puncta. And by tracking the puncta, we can do PIV. We can map the entire velocity of the biofilm. Let me just put it again. Now I want you to notice is that you can see the particle at the edge is moving really fast, and the particle in the middle is stuck, right? Mm-hmm. Okay. So if you measure the velocity profile, you see it's very high at the edge, it's 0 in the center. Why? Because cells have stand up and they don't move in the plate anymore. Okay. So then we can correlate 3 things. One is how vertical cells are. You can see there's a core. This is essentially a Z component of the angle. You have a core that is verticalized, and there's a 0 velocity region where there's no growth in the plane. And accordingly, and also we have this radial organization. Okay, so we can also reproduce this with agent-based simulation. As a control, and we have a strain, which we have a mutant without any surface adhesion, you can see that the velocity profile is just linear, and it has absolutely no radial order. It's completely random. Okay. Now for series, now I have 2 slides of theory. So just wanted to show you how we approach this problem. And so okay, let's just take a step back. So this shows that there is some correlation between the radial organization and this velocity profile. But the question is, how do you show that this is sufficient? So JP at that time came up with this really clever experiment, right? If you start with a string without cell adhesion, we know it's going to be disordered. He drills a hole. We call the hole drilling machine. But essentially, we irradiated the center of the biofilm to kill the cells. Now these cells are not growing anymore anymore. It creates a zero velocity core that allows the biofilms to grow. And let's see what's happening. So we have 1. This is before irradiation. As a control, we know this is going to be disordered. The green color corresponds to the radial organization. We kill the core. That's the core we kill. And magically, cells start to radially organize. We didn't do any genetics. The cells is still not adhered to the surface. What we do know is that what we change is that this is the velocity profile. It's 0 at the center and the highest at the end. Now we kill the core. The velocity is something here and is linear afterwards. That shift is enough to generate a radial organization. Right. So now this we can they force us to think, how can we develop a theory for this radial organization? Or in general, how do we think about cell organization in the biofilm? So the way we treat this is we develop a continuous theory for that. So what we think is that, okay, this is density and alpha is the growth rate. All we are saying is that the growth is actually the density change is driven by growth. And if we avoid, if we just ignore density fluctuation, okay, the gradient of velocity is essentially due to growth. That's fine. The next, we need to couple the growth with the orientation, right? And essentially, Q here is a pneumatic order tensor. We want to see the evolution of the pneumatic order tensor. And what we do know is that if you play a rod-shaped object, in this case a bacterial, in the flow field, if the flow field has a extensional component, we'll try to rotate with it and align with the flow field. But where does the flow field come from here? It comes from the growth of all the cells. Essentially, it's a self-consistent mean theory. The cells grow, divide, it generates a flow field, and as individual cells in this biofilm, I experience this flow field and I align according to it. Is that clear? Right. So that's what we're trying to do. So there's still rotational component, which is not that important. And also, we actually neglect the pneumatic ordering term because when we know that in this mutant, cells are not really pneumatically ordered. So the reason why you see cell-cell parallel alignment isn't because they intrinsically want to align with each other. It's because they're aligning with the flow that have generated. Okay, just to close this, so this growth will also induce stress. At the same, you need something to balance it. That's a friction coefficient with the surface. As the cells expanding, growing, there's some friction. Okay, that's all we need. Okay, then we solve this equation. Well, we first solve it in a case where everybody is growing in 2D. When everybody is growing in 2D, we found that there's no extensional component. There's no driving force for the pneumatic order to align, and that's why a bacterial colony tends to be disordered. Now, the situation is very different if we think about the core. In this core, there's no growth. Alpha in this core is actually 0. And you solve the velocity profile, and you solve the extension component, and miraculously, we see there's an extensional component. So in a 2D radially symmetric case, we can come up with a very simplified equation. And this is all we want to know. Okay, don't mind. Theta here is the deviation of a cell orientation from the radial organization. This is telling you if cells deviate from radial organization, there is some driving force to move it along the radial direction. That's all this is saying. So we confirm that with the agent-based simulation. And the conclusion here is very simple. If you have some radially expanding bacterial colony, there's no driving force to self-organize. But somehow you realize you have a zero velocity core. It can be cells verticalized, or maybe you drill a hole, and then you have some extensional component in the flow field, and the cells to align with it. So this just gives you an example of how we actually go from observation to continuous theory. And I'm glad I can present. Of course, I didn't present to my biology colleagues because it's quite complicated, but just to highlight. But this is a great example to show how we can use observations and trying to develop quantitative theories and explain a biological phenomenon. Just to conclude on this part, we also have the verticalization. And if we also model this process, I'm going to skip the details. But the bottom line is that we can actually model the entire process. The pressure starts to accumulate and is always highest in the middle. That causes cells to verticalize. And this verticalized core starts to expand. That generates a velocity profile, which is low in the center and higher at the edge. That causes cells to radially align. So we have closed the entire circle here. And then why this is interesting for you if you don't really care about bacterial cells, because we figure out that you can essentially use this to explain many other organizations. For example, this is the Drosophila wing development, and this is the plant cells. We figure out that we can use the same series to explain those. And all this is actually summarized in this manuscript. That's the first vignette of the story. Any questions for that?
Yeah, so my understanding, of course, I'm not going to follow the derivation of these equations, but the basic physics is basically a boundary condition problem. Like when you have verticalized the cells in the central region where you have a very firm boundary, cells cannot migrate in.
Yes.
So they can only go outwards. And that's why outside you almost have a free growth condition.
Yes.
And that's enough.
Yeah. And then interesting is that boundary condition is self-proposed is actually imposed by themselves, right? Because the verticalization is also because we didn't set anything. The cells spontaneously develop that, right? Which is a cool thing. And also the boundary condition is changing. As the pressure is increasing, the verticalized region is always keep increasing. Right. It's also a changing boundary condition. So, yeah.
But it's actually a 3D problem, right? I don't know if you have a question that we really solved the 3D problem.
Let's see the 3D problem. Okay. So the second part, everything I talked so far is the 2D. Now let's look from the side. It's just actually a 3D biofilm. So in this case, we're taking advantage of the fact that we're actually putting a gel on top. We can control the gel stiffness by the concentration of the agar. And of course, the stiffer the agar, the flatter the biofilm. That is understandable. But we see a sharp transition. And before that, we have this more hemispherical biofilm. And this is the orientation field because we track individual bacterial cells. And this is the orientation field if you go to very flatter biofilm. So, and if you are studying topological defects, then you immediately realize that the topological structure of the orientation field here and here is very different, right? So, all right, I'll get to that point. So the first thing I want to say is that we have a gel on the top. What's happening at the gel interface? Well, so actually that's not hard to think about. You have some object that is growing, right? As the volume of this biofilm is expanding, the interface between the biofilm and the gel is also expanding. That expansion creates an extension. If you want to think about it, it's umbrella problem, right? So if you think about the surface of the umbrella, of course, it's under very strong tension. And then in our case, the biofilm is growing, that creates the tension. The tension is always in the radial direction. The next question is, do cells care? Cells is growing, they create this tension, but the tension is in the gel, right? Does that transmit back to the cell? And how do we disentangle? Because in the growing biofilm, it's very difficult to disentangle these factors. So, and then at that time, the postdoc Rachel, and she figured out a really nice experiment. But we said, okay, let's not look at biofilm. We'll look at some cells on agarose. And then we'll put an indenter, a spherical indenter that is really small. We indent the agarose gel. When you indent this, you create a radial stretching. Not underneath it, but right here. In here, cells are not in contact with the indenter, but they will feel a radial stretching. And we're trying to see whether they align with the field. Okay? And we first try with a mutant that does not have cell surface adhesion. It doesn't really care. So this is the stretching direction. These are the cell orientation. They're not correlated. And look at this. For the real biofilm cells, when you do this, they feel that there is a string, there's some stretch in the hydrogel. They all align with that direction. Okay, and this is essentially the quantification, right? Without the cell surface adhesion, this is random. With cell surface adhesion, either whether the cell is a cluster or not, they all start to align with the direction of the stretch. So these cell surface adhesion proteins allow the stress or strain in the gel to be transmitted back to itself. Stop here, pause here a little bit, see if that's clear.
Is there a 90-degree rotation? I'm sure when I
understand
it, so it's like.
So this is the stretch. So this is the stretching direction.
Okay.
So this is the direction. Yeah.
So it's like.
So this is rotation. This is the rotation of stretching. But this is the direction of stretching. These cells don't care, and it's just random. Here you stretch, everybody follows that direction. Okay. And that is now we show this is due to these proteins that we now understood how it works. It transmitted stress back. Okay. So. Okay, so you can imagine if this is the what's happening in the gel, and the cells can sense that, cells will also align with that gel. That explains why cells are organized on the top surface. Now is a boundary condition question, right? We know cells will just align with the surface, right? And let's look at these two types of biofilm. One is, of course, taller, and this one is actually fatter. It's actually thinner. But why their topological structure is so different? In this case, you can even see a defect, right? In 2D, it's two defects, but actually it's a defect ring, okay? So the way we rationalize that is this. So in this case, the biofilm is confined or not confined or under a very soft gel. The contact angle here, the effective contact angle here is pretty big. If I have this rod-shaped object, it can smoothly bend through here, and that's the configuration we have. And if you have a biofilm where this contact angle is really small because we have a hard arc on it, the only configuration you can have at the edge is a splay configuration. Okay. Now, if you count the total topological charges in this biofilm, you're gonna have to have some defects inside this biofilm to satisfy the total topological charges, and that's how we rationalize why we see this type of organization. Okay, for those one interested in liquid crystals, and because it's the shape of the cell, I think we can also apply some of these concepts from liquid crystals to this problem.
And do people know that contact angle is much bigger on softer gel? There's some kind of
we measure
deformation.
We measure it. So we measure the contact. Actually, we measure the contact angle for all these different contact conditions. So if you go to the paper, we actually solve the question. We actually predict the contact angle. Because it's a three problem. The biofilm, I can just expand on here. I don't have the slides here. The biofilm wants to grow and expand.
Right.
If you want to, in this configuration, you are deforming the gel. Right? In this configuration, you're not deforming the gel, but you're going between the two interfaces. So there are different energy considerations for these two configurations. And the softer the gel, the easier this configuration goes to, essentially.
So when you solve the problem, you also predict how much deformation.
Yes,
has occurred
and because we have the beads, we have particle tracers in the gel, we can actually map back how much stress it's actually applying.
Yeah.
So I'm skipping a lot of the details here because these are published work. So I want to finish and then talk about some unpublished work. But there's one more thing I want to show you here, which is I find it quite interesting, is because we have this, I showed you a movie of how we actually use the puncta in the bacterial cells to track the flow, right? And that time flow is 2D. And here I just want to show the flow in 3D. Now, interestingly, in soft or unconfined environment, what we call it is a fountain flow, right? It's almost like a fountain, cells go there. Interesting here is that the cells here, because they are moving along the surface, they have a really hard time. So they move really slow, and you have this almost water fountain coming out, okay? And when we have the hard confined, as a consequence, if we look at the cells on the surface, ask whether you always stay on the surface, the answer is no, because a lot of cells are go up and coming back, okay? And on the other hand, when we do this confinement, what we see that the trajectories look like, we call the lawless type, right? If you think about lawless, that's what it look like. And then if you look at now, if you look at this one, all the cells that there are on the surface are pretty much all the time on the surface. So what's going on here? Well, this is still quite unclear, but we are thinking that when the cells are sticky, they have adhesion proteins. When they stick to the interface, right? The interface is actually moving up, and the cells is tracking the gel, so it actually moves together with the gel. So the trajectory points upward instead of bending downwards. So that's a hand-waving argument. We don't have a theory for that, but this is what we see. So this is a really quick way story and if you want to see the detail that we map out, we also include all the derivations in this paper. But what we learned here is that the biofilm is really an active growing material, and as it grows, it actually deforms the environment, and this deformation in the environment can actually go back to the biofilm, influence individual cells, causes a change in the organization and the cell trajectory. So if you want to take this, if you want to take home message, this is what we see. So it's not so different from the tissues, right? We grow tissues on soft substrate, and the cells are moving, and then tissues are moving. They also apply stress to the surface. The type of stress they apply is different, right? We don't have cortical tension, they don't contract, but they need to grow and divide, and that is enough to generate stress and change the environment. Okay. Good? Good so far? Okay. So for the rest of the time, I'm going to tell you some a evolving story that we're planning to wrap up. Now let's think about heterogeneity, right? Everything I showed so far have one color, as if every cell in the biofilm is doing the same thing. But in reality, like microbiologists think that cells have their individuality, just like every one of us here, we're all different. The cells in the biofilm may be very different, and that's something that we want to look into. It turns out that in terms of regulation, there's a single key molecules important for whether a cell decides to go to biofilm or not to make a biofilm. And that is the cyclic di-GMP. Okay? I'm having the molecule structure here. And essentially what it's doing is that if the intracellular concentration of cyclic di-GMP is high, the cells will make biofilm. When the cell concentration is low, the cells will tend to be motile. And this is universally true for all bacteria, not just for Vibrio cholerae. Okay? So in the case of Vibrio cholerae, what we know is that this cyclic di-GMP controls the concentration or control the expression of many major matrix components, including the polysaccharide and the proteins. And all these components, VPS and all those proteins, they're responsible for cell and cell surface adhesion, all the things all the things that are associated with biofilm formation. In the cells, this concentration of this molecule is tightly regulated. There are enzymes that actually synthesize it. There are also enzymes that degrade it. So we're not tracking the enzymes. That's very difficult to do because there are so many of them. What we're doing is that we're tracking the intracellular concentration of the molecule. So I'm going to skip the details. We make a rival switch-based reporter. Essentially, in all the movies you're going to see, if the cells have high level, it will be blue. If it's low level, it will be red. Okay? And blue essentially means the cells is making a lot of matrix. Red just means that it doesn't make matrix. And by we, again, this is done by a current postdoc, Benny Tai, and he's actually on the job market now, who did all the wonderful work that I'm going to show you next. Okay, so enough background. Let's look at what the cells look like. Okay, so the red is a constitutive channel. The cells is all red that allow us to identify them. Blue corresponds to how much matrix each cell is making. And it immediately look like you see this strong heterogeneity. We think about biofilm, we think maybe every cell is hardly working, produce the matrix, everyone can use it. We know that's not true in human society, right? It's also not true in bacterial, right? Some cells are actively making, they're blue here, they're actively making matrix. Some cells just say, I don't care, I'm just in the matrix, but I don't want to do anything. Okay, so we develop tools to actually segment individual cells. And also, again, as a reminder, we use this gel. It's a geometry in which the biofilm is growing between the gel and the glass. Now, by the way, if it's not clear, the gel is infiltrated with all the nutrient molecules and so on and so forth, because the molecules are much smaller than the pore size of the gel. So the cells aren't like can take all the nutrient from the gel. Okay. Okay, I'm going to show you this movie. There are just 2 copies of different colonies. You can focus on either one of them. Okay, there are many things that are going on here in a real biofilm, right? So we went ahead and quantified the intracellular concentration, okay? And on the right is the quantification, on the left is the movie, okay? There are a lot of things that is going on here. The first thing we see is that the concentration overall goes from low to high, the bluer, and then it goes to low again, right? And second, in the middle, during the development, we start to see a bimodal distribution. A population is red, a population is blue. And it seems that what interests us from a physics perspective is that you see this spatial pattern of sorting where the blue cells are mostly in the middle and the red cell is at the edge, right? If you think about eukaryotic cell development, there's differential adhesion theories that cells, if cells have differential tension or differential adhesion, they tend to sort to different parts, okay? But we're seeing the same thing, but is that due to the similar mechanism, right? Well, we first quantify this sorting using a Spearman correlation between the position and the expression level. So essentially, if we have this pattern, and it will be -1. If it's random, it will be 0. So for the Y-type biofilm, we get pretty good sorting, or like a -0.5. And then we ask, what are the physical factors that affect this sorting behavior? We first delete the cell-cell adhesion. Doesn't work, it's still sorted. We also delete the cell surface adhesion protein. It doesn't work, cells even sorted better. When we delete, the thing that really destroys this organization is that if we destroyed both the cell-cell adhesion and cell surface adhesion, essentially this back to E. coli colony. In that case, the fluctuation or the bimodal distribution is still there, but they cannot sort it to the right place. Okay, these are the facts, and then we start to build a model. That's what we do. We make the observation and we try to figure out what's going on. Once we have a qualitative idea, we start to build simulation or theoretical model. Okay, so all right, so this what we see is that sorting requires matrix-mediated adhesion, either cell-cell adhesion or cell surface adhesion. So now we start to think about what is the mechanism of sorting. The biologist tends to think, maybe the cells in the edge in a different environment from the cells at the center. And that's why they respond to the local signals. That's why it works. That's why they have different gene expression pattern. But that cannot be true because we have a mutant, right? We still have edge and center, but it is not sorted. So we come up with a different mechanism. We call it a frictional sorting idea. So what that means? Well, now we know that for some reason cells develop this very heterogeneous expression pattern. And we know like the color we cut here corresponds to how much matrix each cell is producing. Now imagine that if you happen to have a cell that is low cell surface, low cyclodigestion level, then I'm producing less polymers around myself. So I'm less stickier to my neighbor and also less stickier to the surface. And during the expansion of this biofilm colony, then maybe they will be differentially sorted to the edge because there's less resistance to move. Okay, all these complicated things can be simplified in this cartoon. Okay, so like if you have blue cells, you have a lot of sticky around you, and the red ones, they're less sticky. If this is the edge, then you will be raised faster to the edge. Okay, I like this a lot until my postdoc told me this was generated by ChatGPT. So I thought he drew it by hand, but yeah. Anyway, so how do we prove that? So we build agent-based simulation. In agent-based simulation, we assume the cells have two states and a stochastic switching between the two. One is the high frictional state, the last one is the less frictional state. As a movie, and a stochastic switch between the two, which I will show you experimental evidence later, but over time, you start to develop this sorted pattern. Okay? You can change the friction coefficient difference between the two, and you have more and more sorting. Okay? Interestingly, the growth rate, if you assign different growth rate, that doesn't work. It's not about the growth rate, it's really about the physical property change of these two state cell state. Okay? So yeah. So ongoing work, we actually incorporated cell-cell adhesion, and also we simulate this in 3D. But essentially, the basic idea is here. And I just mentioned that we have this model where cells are stochastic switching between the two. So if we want to prove that, we really need to track individual cells in this three-dimensional biofilm, which is technically almost impossible. So we developed this tool that allows us to do it partially. So remember this puncta cell, right? We use this to track the flow field. But now what we're doing is that we also put this fluorescent reporter on it, okay? But what we're doing is that we image these, the puncta very regularly so we can track the puncta. And from time to time, we image the cell so that we avoid photobleaching problem. And we know each puncta has to belong to a cell, right? The puncta is inside the cell. So this allows to not only track, we can use this to track the puncta as a way to track individual cells. And importantly, that allows us to track the expression level, in this case, the secrete GMP. But think about technology-wise, this allows to follow individual cells in 3 dimensions for any genes that you want to track. Okay? Here's what you see. So this is the cell that starts from here, and we can follow it through the entire developmental process. Okay? If you want to think about cell state transition, this is nightmare, right? It starts from high secrete GMP, it goes low, goes high again. So bacterial cells don't decide, oh, this is the pathway I want to go to. It is undecided. It's actually stochastically switching between different states. And this is the expression level. And here's the expression level. And now it became really clear, right? When the expression level is low, I'm not making matrix. Now I start to accelerate because I'm less sticky. There's also a time delay, right? It takes some time for me to start move. And when it goes low, when the cycle that GP goes high again, I stop because I'm stickier to the surface and my neighbor. Okay? So just to show you some example, we have a case where cells is low cycle energy, keep moving, and somehow stochastically switch to the high state and stays there. Okay? And I have the opposite example. Might take a while. The cells is actually blue. It's pretty sticky. And at some point, okay, just look at that cell. Now it turns red. This one. Now it turns red. Boom, it starts to shoot off. Okay, so this interesting so now this really allows us to correlate the expression level in the cell to the phenotype, to the behavior of individual cells in the biofilm. Okay, question?
So you just get the Z component from the position of the puncta, basically?
This is actually 3D tracking, so we have I'm showing a 2D, I'm showing mostly what's happening in 2D. So actually we track this entire 3D.
Okay.
But sorry, when this guy turns red, start moving, are they actually motile? They're just being pushed.
It's not motile, and nothing here is motile.
So they're being pushed by the.
So nothing here is motile. There's no actual flow fluid, and when they say move or float, it's all material flow. So remember, in the first part, we treat the growing biofilm as a fountain or as a source, and it's a flow. It's a flow. It's essentially if you imagine it's expansion, right? If you have a fountain, you are pumping water into this, these are grow, right? Now you pump water, but I have two materials, they have different properties. They will be actually moved in different speed. That's what it's saying.
Yeah. So the quote unquote motile cells aren't really motile.
They're not motile.
They just don't have the sticky stuff.
Yeah. Because they don't have matrix around it.
Mm-hmm.
Okay.
But is there any cells actually changing back from the red to the blue?
Yeah. So like the back to blue. So that this example is a cell that going back and forth undecided. Okay. So. All right, the last experimental thing we'll show is here. And because we have this radial organization which I explained in the first part, now and that actually gives you the lineage, right? If you have a cells is red, then all the dotted cells will be red. If it's blue, it will be blue for a while. And then we know the red cells is less stickier, which means this lane, it will be less stickier. So we call it fast lanes. Some of you in the audience will know what I'm talking about, because it reminds you of the anyway, so these red lanes correspond to the fast lanes because they're less sticky. They're almost like a pumping cells through these lanes. So you can imagine, you can see here, this is the cyclic This is the level, but now it's across the circumferences. Okay? Right. Okay. So I think so the last thing we talk about, why does it matter? We need to ask this question. Or maybe the question is that if this is so obvious, why haven't people seen this before? So we repeat this experiment in an unconfined biofilm, right? Nothing to confine them. And we see that cells will grow, expand, always blue until every bit turns red. Okay? That's surprising, and also that we get frustrated. Okay, maybe what we see is artifact because we're growing this geometry, until we realize that it's essentially a survival bias. You look at the biofilm, right? You look at what is actually staying on surface. Yes, everybody will be high psychedelic GP and produce matrix, because the one that will turn red, the one that's decided not to make biofilm, will be gone between the two imaging steps. So how do we test this? Well, if we remove the motility genes, the cells on the delta polymer means they are not motile, then we are able to partly recover this sorting pattern. Okay, you can start to see the cells, the red cells around the blue core. So, and the last thing I want to show you is that think about what's the point of cells to fluctuate between two states, right? What's good about it, right? So we did an experiment, we did a competition experiment where we have a Y-type cell that is able to switch between different states. We have cells that are locked in one state, either the low one or high one. And our hypothesis is that maybe this is good for cells to be in the fluctuating environment, in an uncertain environment. The way we do this is that we grow them in a flow chamber for a short period of time, and we use the effluent of the first chamber to inoculate the second chamber, and then so on and so forth. That is stimulating the effect of a flush. Maybe this is a niche, for example, a food particle, and it goes to another particle, goes to another food particle. So when we do this, we realize that over time, you start to see in the final chip here, we see a concentrating effect of the Y-type cells. If you quantify them over time, you start to see that the Y-type cells, the abundance of the Y-type cells is increasing. Why? Because the Y-type cell is able to grow some biofilm here, accumulate biomass, but it's also able to disperse those. It's also able to go from the first chamber to the second chamber because it's able to switch between the two lifestyles, and eventually it goes to colonize the next one and next one. You can imagine that in nature, this process has been repeated millions of times, where you colonize some places for a little bit, and go to the next, go to the next. The winner isn't the one that's stuck in a particular state. The winner is the one that can fluctuate between the two states. Okay? So,
How did the experiment go exactly? You have like pipes connecting these three devices continuously? Or
we first grow this for 6 hours to accumulate that. Then we connected the outwarding pipe to a fresh chamber.
Okay, and then flow for some time.
And yeah, very quickly.
Okay.
Because the chamber volume is pretty small. Then the cells start to grow this. We repeat it 6 hours. You can do it. You can keep doing this. So, okay. So if you keep doing it, if you keep like in this biofilm case, then the only biofilm forming will be waning, right?
Yeah.
But if you do this fluctuating environment, you see that what helps is that form a little bit of biofilm, but don't commit, and later go to the next chamber, so on and so forth. So I think that goes to the conclusion here for this part, and my talk is cells somehow they spontaneously develop heterogeneity, almost bimodal distribution in a growing biofilm. And this is in contrast to a textbook example. So if you open any textbook on biofilm, you will see this picture. Cells go to the form biofilm and coordinatedly detach from the surface when nutrient is limited. What we are saying is that, no, cells are never committed collectively into one state. They're always trying to figure out what is going on. And these low psychedelic GP cells, because of their less sticky, they will physically sorted to the periphery, and that's where the place they can actually leave the biofilm. And the punchline here is that a bacterial population never 100% commit to the biofilm state. It's probably what we call a bad hedging behavior, because yes, I'm now eating food on this surface, but maybe there's something good thing out there. I don't want to fully commit. Some population of cells will will start to move outside the colony and try to find new places to colonize. And that's what we're thinking is going on. So it's very different from the development of tissues of us, where cells committed. Correct me if I'm wrong. So if cells are committed to a certain state and just go that pathway. For bacterial cells, the things tends to be less deterministic. So that's all my talk. I'd like to thank my labs. That's a movie. That's the last movie I want to show. Okay, and then actually in front of my house, and this, particularly both the work that, especially the second part of my work, the third part of my work is done by Benny, who's, let's see, here, hiding here. So yeah, happy to take any questions.
I was actually wondering the blob of all the cells, they're kind of like they're covered with the gel that provides the nutrients.
Yeah.
I'm wondering how much effect does that have, because you can only get the nutrients from the interface of the thing, right, in the 3-dimensional. As long as if it's like a big blob, like
it's the nutrients come everywhere. So we have a nutrient actually above the gel. There's a full reservoir of nutrients.
Right.
Yeah.
But the ones who are the ones which are sort of localized inside the blob.
Yes.
They should be like getting less nutrients.
Correct.
Right?
So the nutrient can diffuse to a certain extent into the biofilm,
right.
But there is we call it diffusion length, or let's say starvation zone, right? There's people have shown that the diffusion is about 50 micron. Once then you start to have diffusion problem into it.
Okay.
So we tend to study biofilm that are smaller than that.
Okay.
So
yeah.
But overall, later on you see that the cyclodide GP level turns off. Like everybody goes to red because they run into nutrient problem collected. And also it's a very thin biofilm, right? We're not talking about diffusion from the outside, the diffusion from the top. But that's a great question. Thank you.
So this blue to red change is nutrient dependent, like
the blue
depletion of nutrients.
There's two things. One, I think at first it's stochastic.
Okay.
And second is nutrient dependent, also quorum sensing dependent. So I didn't talk about that part. So bacterial cells also change their gene expression depending on the cell density, right? They can sense how many neighbors they are, and what we have shown that in the quorum sensing mutant, this will be very different. In particular, for Vibrio cholerae, the more as the cells feel, oh, there are a lot of cells around me, probably this is not a place to stay. They will actually go. So there's also, as this biofilm is growing, there are more and more neighbors. Mm-hmm. They also tend to lower the cyclodide G level. So that there's never one single factors there, right? Mm-hmm. There's nutrient, there's cell density, there's stochastic switching, all together define the development.
Good. That's a lot of questions already, but are there more questions? Okay, if not, then thanks for speaking.
Okay. Thank you.
You have some of this stuff in your lab?
No, we don't work with Vibrio.
You have pseudomonas. At least you have the other two stuff.
Yeah, we have pseudomonas original.
But it's not harmful, right?
It could be harmful.
It's the same safety level as
if your student makes a mistake, will we all be in danger?
If they drink it, they could get sick.
Drink it, yeah.
If they get sick, the chance of spreading to other people is very slim. It's not like COVID.
So what? This is a bacteria, right? Why is the spread can be so strong compared to virus? Can be airborne, right? But the bacteria is not airborne.
So this one is it's waterborne. So it's through, that's why like in Haiti or in other places where there's a wall that destroys the water purification.
Or maybe I just overlook it.
So you have a source and
make sure
spread,
okay.
That's how it works. But here we don't, not so much in the US because we have clean water provision. Okay. That's not true. Unfortunately, there are walls.
All right, to look at on your side, send maybe I overlooked it.
Okay, yeah, I'll just send it to you.
Oh, thank you for doing that.
Okay, yeah, no problem. Happy to
Don't delete on your side. If I don't get it,
okay.
Just after you. Okay, so I will be away next week. After that, I should try to get better on meeting with you guys. Probably I'll start meeting with you then.
Okay.
He's only going to be here for a couple of months.
Professor Costello.
Nice to meet you.
Nice to meet you.
Thank you for
Sounds good.
Good talk.
Okay, thank you.
I'll see you. What time in restaurant?
5:30.
5:30.
Okay. Are you just going to go down your?
No, I have a student to meet right now.
Okay.
And then
Right, go
we'll
meet the
go down
student
there.
now.
Yeah.
Okay.
I had one question. How universal is this behavior? I mean, in the sense that,
Which behavior?
I mean, this sort of growth you're seeing here of the biofilm. I mean, if you fiddle
Which
about,
part?
well, any of it. I mean, if you take some complete generic model of,
Your student. Wait for you.
I mean, is it sensitive to the parameters in your model?
Good question. So this is a model organism we use, so I can only comment on it. But I think it's pretty the growth. The cyclotide GP we have, I think is pretty universal because people know that this is a single molecule that controls all the behavior. The organization, we haven't tested in other bacteria, but there are also some evidence. I think that even in E. coli, if you can say people have seen this vertical organization and the radial organization.
Yeah, it might be rather common.
It's rather common. People sometimes you don't have the resolution to track the development. When you look at, and the people with the newer development, people are looking at more and more different species, and then they are reports of similar organization. So we're just I think sometimes you just don't know if you don't look at it.
Because in biologists, right, they have their own, like he's recognized as a cholera guy. He studied this bacterium, and then there are millions, multi-million-dollar NIH money is invested. Okay, but then there's not going to be multi-million dollars for some other obscure, not as important bacteria. Wow. So biologists, they hate generalization. So you can't just say, you can speculate, but I think it's pretty general phenomenon. Like we study
the equation we wrote down is general.
The mechanical explanation, but I mean just how many bacteria species behave like that, right?
I
Probably
think
a lot.
It depends on whether you really spend time and energy to look into it.
Right.
Yeah.
And most people don't have money to spend time and energy to do it, because this thing takes like tons of hours to do it. Experience all these things.
Or also a lot. We are benefiting on a lot of genetic tools. For example, we need fluorescent proteins.
Yeah.
So you need to be able to genetically manipulate the bacteria, right? That's not true for some. Like if you randomly give me a species, maybe that's not true. But if you can do it, you probably will see this is actually universal. Does that make sense? Yeah. Depends on whether you look at it.
That's like most biologists, microbiologists study E. coli. Why? Because everybody studies it. You have all the genes, all the tools.
Right.
But then you have, fewer number, but still cholera is so important, so you have a bunch of people who study that. So he has transformed himself to be one of the cholera guys.
Well, but you see, the approach we use is still physics. The approach we use, right, is also physics-based. It's a main field theory.
But most people who do this, right, never gone through like college physics, so they don't they don't have this type of training.
Right.
So they have all the gene stuff. So he goes through the physics training.
But it's fair enough that. But I would still like to answer the question. Look, okay, E. coli say do this sort of thing.
Mm-hmm.
What about other bacteria, or is this sort of thing special to E. coli?
Yeah.
If it is special to E. coli, why?
Yeah. So I can see that. So for example, I stress the importance of cell surface adhesion, and they need to be able to generate the molecule for them to stick to the surface. E. coli does not do that, but some other species may do that, but they're using different molecules. What I'm saying is that as long as you have the molecule and you achieve this physical parameter or stickiness, you will see this. But what is not universal is that you may make a different molecule, allows you to stick to the surface.
Yeah.
But here we're saying but what is universal is what's the consequence or the physics after that.
Yeah. So the philosophy here is basically saying biology works, it's material behavior, controls biological behavior.
Mm-hmm.
And then different species may use different material, different specific proteins, because there are like God knows how many hundreds, thousands of them.
Okay, now suppose you just change the substrate.
Okay. Yeah.
What happens then? Does it the same sort of behavior, or does it behave differently?
Yeah. So that's a good question. So we haven't done too much along that line.
Right.
So you could use another model of bacteria species with using different proteins. As long as the protein, make them adhere to each other, the prediction will probably be similar behavior.
Right.
But then if you change the substrate, that you are changing when you change the substrate, you're changing maybe the boundary conditions. You're changing it. That could be the change. But that's something that we can use physics to still understand.
Yeah. So biologists go for specificity. Physicists, you always want to have the bigger picture. But I think the material property dictates actual biological phenomena is important. it's important
And
to
what,
show.
what I'm trying to say is that also you can use the equations to describe this. That's what I like. For some project, we're able to develop a continuous theory and compare and test it again in the experiment, which I found it to be the most pleasing part.
Yeah.
Oftentimes we can come up with model, but it's really hard to experimentally test it. But this we have a very quantifiable model to do. And sometimes we have a biological phenomenon which then it becomes too complicated to boil down to a few lines of equations.
Yeah.
But here we're trying to find a compromise where we can do both.
You can do both, yeah. But most of the microbiologists, they don't really, they close their eyes when you write these equations. What really their view important, they cannot deny actual experimental facts. So even some of those facts, it's unique that his lab did. Because most people just assume, okay, they grow biofilm, after a few days, you have these big cavities. I've seen with pseudomonas, people show that. How did that happen? We don't know. They just have confocal images of, 5-day-old biofilms, 7-day biofilms, like 9-day biofilms. Show, oh, there's this big cavity. There's a lot of swimming cells. And they don't talk about what's going on. Here, they actually show the process over time of cells switching between the two sticky and non-sticky, and they actively go away. So this kind of actual information is valuable because biologists that go about facts, right? They don't follow logic. If you say some logic, they like that.
Well,
show me the
I
data.
think that there's part you show the phenomenon, and that's always is a fact, right? Whether you develop a theory, whether they buy the theory or they care about the theory is a different question. But that's what we love to as a transformer trading software fit. That's what we want to do.
Yeah.
But that may be just a fun thing to do.
At least it doesn't hurt.
It doesn't hurt.
For them, they don't play with logic. We have some things we show cells divide, right? They get over time. They get from longer lengths to more and shorter lengths. And I said they have divided. They said, show me the data. Show the cell actually dividing. Why do you need to show that? At one moment, these cells are longer, next moment they're shorter. Isn't that obvious? They have divided into two. They want to see the data. We have the movie to show, like they catch them in action. Look, these cells has now forming this puncture.
The first part, the first two stories are actually published. So the first two papers are published. If you go to the
so they're called septa.
If you go to my website and find the first also the NIJ.
And you have to show them in action. Like fluorescent label. Look, now this at this moment. It's very quick. Very quick. So like we have to show them those data for them to
But if you want to see
justify what we say.
No.
So it's really different to emphasize. The biology gives them credit. They emphasize on data. Sometimes it's stupid data. Because you spend, that's why their graduate students spend like 7-8 years to get their data.
Of course, they emphasize, they concentrate on the data.
I can show the